Alright, I know we're all very tired from the drawing pad and sketching and the crosses, so today we're going to be looking at the exceptions to Mendel's rules. Yes, there were laws, and they generally do apply. However, there are still some rare cases where these rules, these laws, they are no longer in place because we have some sort of weird stuff that's going on. So, they recall that these were the Mendel's rules. We had law of segregation, that all alleles will separate into gametes, and each gamete will carry one allele. And remember that you are formed by a sperm cell and an egg cell, one from dad, one from mom. So you have two copies of the trait for the same, two copies of the allele for the same trait. So for example, let's say you're describing, okay, let's do plants. For example, plants, it could either be yellow or green. So the mom was pure yellow and the dad was pure green. So the dad passed the pure green gene and the mom passed the pure yellow gene, but yellow was dominant against green. So the child is going to be a heterozygote. However, the child's going to be yellow because it got completely masked. So that was the law of segregation. And independent assortment just states that the chance of inheriting one trait does not impact the chance of inheriting another trait, which is pretty simple. Law of dominance shows that some trait is dominant over the others, causing the recessive ones to be masked completely, and they have a better chance of showing in the next generation. So for example, your mom and your dad reproduce to create you. However, your mom, or maybe let's just say somebody, their mom or their dad could potentially had a recessive disease. However, because their mom was a homozygous no disease, like they were completely healthy, so you actually are a carrier of this potential disease. However, it doesn't get shown because it's a recessive disease. But when you reproduce, you have a 50% chance of passing that, disease gene. So there's a way better chance of showing it in the next generation. So recessive genes, they hide. However, so in the first case we're going to be looking at is incomplete dominance. The phenotype of the heterozygote is between the homozygote. So right here we have a true breeding or homozygous red plant or flower, and they crossed with a homozygous white plant. However, when they mix together, they create this pink plant. So they merge to create a different color, and that's incomplete dominance. Codominance, on the other hand, is that both traits are expressed, and they're not expressed in like another color. They're purely expressed already in their very own way. So incomplete dominance is you're creating some sort of new stuff, while codominance, you're using the stuff that you already have. So both alleles are fully expressed. So right here we have pure red, true breeding, homozygote red flower crossed with a homozygote white flower to create red flowers with with a little bit of white flowers on there, or white flowers with a little bit of red on there. So it depends on the dominancy. So the other thing we're going to look at is multiple alleles. So more than two variations exist for one trait. So for example, we have on the right the rabbits. So they could have all of these different types of alleles. The C is brown, the CCH is chinchilla, and the CH is Himalayan, and the C is completely white. So usually this is the most dominant because it's a capital letter, and then we add on to it. Dominance, this is supposed to be more dominant to this, and then you add a letter here, and this because it says lowercase just there. So usually I will put this as the most dominant, second dominant, third dominant, and fourth dominant. I don't know why they arranged it in that weird way. But you usually add more letters when you go on. So first you see C, that's sure, it's dominant. And then you'll have a lowercase c, and we always learn that capital is dominant over lower. So the lower c is going to be recessive. And then you have these, they're recessive to this, recessive to that. But however, this is dominant to this trait right here. However, more than two variations exist does not mean this does not mean that you escape the law of segregation. You're still gonna get two copies of alleles for that same trait. You're going to have a CC. Sure, it's full brown, it's full chinchilla, it's full Himalaya, it's pure albino, whatever. You're still going to get one from dad, one from mom. This still exists. This still exists here. So you gotta know that the laws, when you have these exceptions, maybe only one of them is broken. However, it's still considered an exception. So here, this, you just need to know, this is just more color or variation. You still only have 2 alleles per trait. I want you to remember, multiple alleles means you have multiple variation and possibilities, but you still only have 2 possible alleles for each trait. Now we're going to talk about polygenic inheritance. More than 2 alleles for each trait. In humans, our skin color, eye color, height, a bunch of other stuff all depends on a bunch of alleles. So you can see on the right, we have AABBCC, capital AABBCC. So they arrange around like this. But now, hold up. Listen to me. On the left, the only you are going to take 1. 1A, 1B, 1C. And on the right, it's the same thing. 1A, 1B, 1C. So you look at the possible combinations. On the left, you have lowercase a, lowercase b, lowercase c, and that's the only possible combination. And for the right, it's the same thing. Only capital case A, capital B, capital C. However, when you look at here, it's crossed with itself. So big A, small a, big B, small b, big C, small c. So that is supposed to give you a good amount of variation. So for example, you could have big A, big B, big C, big A, small b, big C, big A, big B, small c, and all of that. So remember, so when you're doing all of these polygenic inheritance, I want you to ignore the BBCC first. So only look at A. Is the dad going to be able to, which A is the dad going to be passing on, and which B is the dad going to be passing on? And because of this, all of this pure mixing and genetic variation and all of this, we are crossed, and we will always get this distribution. This distribution right here, it tells us that the middle is going to be the largest. The average will always be the most. The most common will always be the average. That's why it's the average. Alright? So it's really rare. It's extremely, extremely rare that you're going to be able to get a full, very light-skinned kid from this. It's very rare. But there's a possibility, but it's very rare. There is just possibility that's if the dad just somehow passed the little a, little b, little c, and the mom passed the little a, little b, little c, that's a possibility. And on top of this, there's a chance that they could pass a big A, big B, big C, and big A, big B, big C, that 2 less toned skin somehow just have a really dark skinned child. So you have a 1 out of 64% chance, 1 out of 64 chance of actually getting one of these. So it's really rare, but it still exists. However, you're just going to remember, always, 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 the center is going to be the most common, and it's going to be the average. Now, there are external factors on this as well. Yes, genotypes, and they dictate kind of how you start with. However, that's not it. It dictates how you start with, but things they will eventually modify, and you will start evolving and all of that stuff. So for example, in a flower that's normally grown in a pH level of 7 to 8 or whatever, sure, it's going to be pretty healthy, and it's going to grow to the same color that it's supposed to be, that the genotype tells it to. However, when that flower is placed in an abnormal environment, a higher pH or a lower pH, or there could be other stuff in play, like let's say there's some sort of chemicals going around, the flower color, it's going to change. On the right, we have the inherited genotypes, and then you could have all of these effects. Viruses, bacteria, that's fish, I think, antioxidants, prebiotics, folate, allergens, and pollutants. So they could potentially expose this genotype to some sort of, pollution, and the genotype is not going to function the way it normally works. And then you have environmental exposure, which modify gene expression. You could have tissue effects, immune effects, and all of that. And finally, you're going to come out with this evolved phenotype. So, especially for humans, we're such complex animals. So when we look at these stuff, these usually, they don't really resemble us. The genotype does not completely resemble you, essentially. You could have some variation going around here because you're so much different. And that's about it for all of this variation. So we're going to be doing some of these questions very soon, and hopefully, just hopefully, we'll be able to teach you some codominance and incomplete dominance crossings. All right, so let's go back to the class. Today we're going to be introducing more exceptions. So we're going to look at the first one, the idea of lethal genes. So in the monohybrid and dihybrid crosses, we usually have a set ratio for the set amount of input. So for example, if I have two heterozygotes in a dihybrid cross, they will always yield a phenotype ratio of 9331. However, when we input this idea of lethal genes, even though if both of them are heterozygote, we have the phenotype ratio. It's going to be mixed up. Because the lethal genes, there are genes that could potentially kill the individual. They could be dominant, recessive, or conditional. And when we do a cross that has lethal genes, and let's say we have a recessive lethal gene. So when we see that individual, we automatically cross him out of the monohybrid cross. Why? Because he's already dead. So I have a couple of examples of lethal genes. So obviously we have Huntington's disease. That is a dominant lethal gene. So the symptom of Huntington's disease actually appear much later in life. So it kind of allows the gene to be passed on to its offspring. So what does that mean? It means that these individuals, they're able to pass the gene on. However, they won't be classified in our monohybrid because they're going to be dead. And we just assume that most likely that they are unable to pass it on. And there are also recessive lethal disease, and I could give you like a cystic fibrosis. So there are two copies of that allele, which is needed to cause a lethal effect. And when we do a monohybrid cross, we just literally cross it out. And we're gonna talk also about the idea of heterozygous advantage. So usually when you are a carrier, you're considered lucky because hey, you didn't get the disease. You're one allele from getting that disease. However, these heterozygotes, they're more better than you think. So there are situations where the carrier of certain disease is actually more fit than the homozygous dominant or homozygous recessive, basically meaning that sometimes when you carry some sort of disease, it might even give you a benefit than the ones that don't have it at all. Example could be sickle cell anemia. So when you have a normal red blood cell, let's say you're able to carry 10 oxygen, and a full sickle cell anemia. So sickle cell anemia is a recessive disease for information. So when you have full sickle cell anemia, your red blood cells cannot carry 10. It can only probably carry around 4, maybe even 5. However, when you are a carrier, your red blood cells, they half look like a normal one, and they half look like a sickle cell anemia, meaning they will be somewhere in the middle. They maybe be able to carry 7 or 8 oxygen. So in that case, we have another disease called malaria. So malaria it's pretty common in places like Southeast Asia or like more to the southwestern Asia somewhere. So it's pretty common there, and it targets healthy red blood cells. So if you're somehow a modified version of this red blood cells, you are a carrier of sickle cell anemia, you're actually completely immune to this disease. So therefore, you have a heterozygote advantage. So we're also going to be looking at the idea of X-linked. So funny, some of the diseases, they could potentially be X-linked. So humans, we all have XY or XX chromosomes, which dictates our biological gender. And I put biological because I want to make everyone feel included. You know, if you identify as something else, sure, whatever. So since males and females, we have different XY chromosomes. Females have XX and males we have XY. It means that the chance of inheriting these X-linked disease is actually different. This means that there are some unique inheritance patterns. And do I have a next slide for that? Yes, I do. So hemophilia. It's a disorder that affects blood clotting. There could be red and green color blindness. affects the ability to distinguish between red and green colors, and DMD, which is a muscle wasting condition causing your muscle to go sore and numb. So all of these diseases, they actually have a greater chance of affecting male. Well, why? On the left, you can see if the father is fully unaffected and the mother is a carrier, then the daughters are going to be completely safe. Because dad will, when you have a daughter, dad cannot pass on his Y gene because Y genes are for male and male only. So dad will always pass his X gene on to everybody, no matter who it is. Sorry, to the girls, to the females, yeah. But to the boys, dad will always pass the Y gene. So let's just look at this first. So when dad passes the X gene to the female, to the daughter, it means that the daughter will 100% not have that disease, but it could be a carrier. However, when the dad passes the X to the son, it means that the son will 50% to be a carrier. Sorry, it will be 50% to be a carrier or 50% to be completely healthy. And when I say carrier for a son, you should realize that dad already passed on the Y gene. So there are no more things to cover it. So if this disease, no matter if it's dominant or recessive, you only have one copy that's dictating this disease. So if mom somehow passed this X-linked recessive allele to that son is going to die. Or, well, not die, but she's going to have the disease. Or if mom somehow passes the healthy gene, the son is going to be healthy. So essentially it means that the daughter, if that is completely unaffected, the daughter will for sure not have the disease. They could be a carrier, but they will not have the disease. But the son, that's a 50-50 right there. You're playing some games, guys. It's not fun. And let's say you have dad is affected. Let's say dad has the disease. So for the daughters, they will 100% be a carrier. They will 100% be a carrier, but the son is completely safe. However, usually, your mom is likely going to be unaffected. So because females has 2XX chromosome, meaning that they have more possibility of being a carrier than someone actually expressing the disease. However, because some have an XY, it generally means that the son, if your mom has a disease, you're pretty much GG's. You're playing a 50-50 here. So the son will always, the son cannot be a carrier. First, you gotta understand. So the son can never be a carrier, no matter if it's dominant or recessive. As long as it's an X-linked disease, the son can never be a carrier because the son will always receive the Y from his dad, and whether he receives the healthy from mom or the unhealthy from mom, it's all going to express. So that means, with that said, females have more, abilities to make up for the wrong. So if somehow this female had this she become a carrier because of her dad, her mom still has a good chance of passing the healthy gene to her. But however, if a son got this unhealthy gene from his mom, then his dad can't save him anymore. Now we're going to look at something different. Well, you see, we have blood types. All humans does. We all have blood types. You're either type A, O, B. Okay, A, B, AB, or O. So these can be written as homozygous or heterozygous. So here we have IAIA. I is just blood type. You just write it like that. IAIA, IBIB, IAIB, or II. The small I, lowercase I, is for O. However, you could also be a heterozygous. You could be a IAI, IBI, or IAIB because this is already a heterozygous, or just double I because blood O is recessive to all of these, to AB because it can't fit anywhere in here. So how does this work? Well, type A blood has A antigens and anti-B antigens, which will essentially kill blood type B. So if you give a type B blood to a type A person that needs blood right now, they're not going to be able to use it because their body will be able to be like, oh, there are some B antigens here. I'm going to kill all of these. So he kills all that. And the B, they will, and vice versa, obviously. When you give a B type blood, some A blood, they're going to be like, oh, what the hell is this? I'm not taking it. And O has this special thing that it cannot take any blood from A or B because they have these antigens. The O person will die. So with that said, we're going to look at the RH factor. So a blood type could be positive or negative. And remember, positive is dominant against negative. So when a blood type is positive, it will have RH antigens, meaning that negative type blood cannot receive positive blood types. So these RH antigens, they will start killing the blood if you put them into a negative blood. However, because of a negative blood types, they don't have these RH antigens. They're able to give blood to everyone. So what does that mean? It means that negative blood can donate to positive blood since RH is absent. However, positive blood cannot donate back. So let's look at this. Blood donation. AB can receive blood from both type A, type A, type B, and type O. AB can receive it from everyone. And when we go further to break it down to AB positive or negative, AB positive can receive blood from A positive, A negative, B positive, B negative, O positive, O negative. However, AB minus can only receive blood from A minus, B minus, or O minus. A can only receive blood from A and O. And break it further, A plus, A positive can receive blood from A positive, A negative, O positive, O negative. And A negative can only receive blood from A negative, O negative. And B can receive, B is the same as A, so we don't talk about it. But O can only receive blood from O. O plus, O positive can only receive blood from O positive or O negative. However, O negative, that's the bad one. If you're O negative and you somehow got into a real big tragedy and you're losing a lot of blood, unfortunately for you, you can only receive blood from O negative. However, because you are O negative, you don't have any antigens. You don't have A, you don't have B, you don't have RH or anything. You're able to actually donate blood to anyone. And that's why O negative is often referred to as the universal panda blood. Because it's just rare. And I'm pretty sure they have these foundations where they connect all of these people together so that, if somebody that's a O negative, they got into some sort of big trouble or a car accident and they start losing a lot of blood, they can quickly connect them and be like, oh, come, there's somebody in need of blood, they're dying. Alright, so that is pretty much it that I have to talk about. We're going to do a couple examples that involves these type of stuff. So let me pull that up real quick. Sketchpad. Perfect. So we're gonna go ahead and do some examples here because I feel like we need to. So let's start. I will only draw it out for you, and I will give you the let statement and all that. I won't do it exactly for you because it's gonna look real messy and stuff. Whatever. Alright, so let's look. Let's see. Hemophilia is an X-linked recessive disease. The blood in hemophilia does not clot properly. A man has hemophilia. What are the chances that he will pass hemophilia allele to his children? From which parent did he get the hemophilia allele? Alright, so a man, alright, what does a man have? A man has XY. Now, remember, since male and male cannot have babies yet, and neither can females and females, it means that he got the Y from his dad. And remember, you only get one from your dad, one from your mom. So where did he get the X from? Mom. And this is a bad disease. So let's set up the let statement first, actually. That's supposed to be the first step. Let X, let's say H, be hemophilia. And guys, the H is supposed to be a subscript, and let H be non-hemophilia, because remember, it's recessive. So it's supposed to be actually written like this. Let me just write it for you. So it's actually supposed to be written like this. X, H, that, or X, H. That's how you were supposed to write it, but whatever. So first we answer the question B. Where did he get it from? Well, because he got the Y from dad, so it meant he got this. this bad gene, this negative, this small h, he got it from his mom. So B, we're gonna answer B. Okay, let's put a text box here. B. His mother passed him the hemophilia gene. Now let's look at the question number A, if that's possible. So let's see. What are the chances that he will pass this hemophilia allele to his children? So I do this a little funnily, but let's see. H, Y. Now let's do a theoretical cross. So obviously he have, okay, so if we only look at him, okay, we only look at him, so we gotta look at this again. So if it's a male, he's always going to pass the Y gene for a son. However, for a daughter, which I will write DAU, daughter, he will 100% pass his X gene. So it means that he has let's just assume that the chance of getting a son or daughter is 50%. So if you get a son, he's gonna pass a Y. If you get a daughter, he's gonna pass an X. It means that he has a 50% chance of passing hemophilia allele to his children. And that was the first X-linked cross question that you ever done, I hope. If you did it before, then I'm happy for you. If you didn't, that's okay. But whatever. Now you know. So let's do another thing about codominance, because codominance is pretty fun. Let's see. All right. Pure red flowers are crossed with pure white flowers. So let's do the let statement. Let R be red and let W be white. So you gotta be able to identify what's going on here, because hey, they didn't tell you the dominancy, so you gotta know this is codominance, because hey, you know. Oh, wait, this is incomplete dominance, is it not? Yeah, it's incomplete dominance. So let's see. What conclusions can you draw about color? Okay, so let's just write it here. This is incomplete dominance since you are mixing two types of genes and creating a new color. So whenever you create something new, that that is incomplete dominance. If the F1s were crossed, okay, so obviously the genotype, okay, no, hold up. The genotype of the first offspring was RW, right? Because the dad was RR, pure red and pure white. So when they cross, this is gonna pass the R gene on, and this is going to pass the W gene on. So obviously you come here and come here. So it has RW. So let's see, RW crosses with RW. So it could pass the R gene or the W gene. Now what's gonna happen? So RW, RW. Remember, this is a monocross. R W W W R W. All right. So what does that mean? If F1's what ratios of colors? Right. Now let's type it in here. So we gotta identify here what's going on. So we see that there is one homozygous red. There is. So let's just identify the ratio first. This is 1, 2, and 1. All right. So this is one type of genotype, one type of genotype, one type of genotype. The ratio is 1 to 2 to 1. And it is pure red, pink, and pure white respectively. So that is apparently how you do incomplete dominance, because you just cross it. It's usually pretty simple. Let's see codominance. Do I have a codominance example? Actually, no. Codominance are pretty much the same thing. Let's do some question called multiple alleles, if you remember. Okay. Multiple alleles. Yellow coat color is produced by a homozygous genotype G1 G1. Cream color is oh wait, no, that's codominance again. But hold up. We'll do this. Why not? We'll do this. So Yellow is CYCY, cream is CYCW, white is CWCW. What genotype ratio between cream color are mating between cream color individual likely going to produce? So cream color was heterozygote, guys. So remember. So I'm just gonna show you the cross. It's going to be CYCW crosses with CYCW. Now what will that yield? That will basically yield us the same thing here. Because here, let me show you guys the cross. WCY. No, I wrote a little too big. It's okay, whatever. So I'll just write this because why not? YY, YW, WW, YW. So remember, this goes to here, and that goes there. So that's how you do it. So essentially, you have two cream color, one pure yellow, and one pure white. So these questions shouldn't take you that long on the test, by the way. All right, let's do codominance. Yeah, let's do codominance. Why not? All right. So the palmetto horse is a hybrid exhibiting a golden color with lightened mane and tail. A pair of codominant alleles, D1, D2, known to be involved in inheritance. Genotypes of the homozygous D1 allele are chestnut. Heterozygote are palmetto in color, and from matings between. Okay. So I told you guys, it's basically the same way you approach it. So remember, you're mating. Oh, wow, wow, wow. I don't like this, buddy. Give me that. So you're mating between a D1, D2, X. I'm not talking about your basketball or your sports league, but I hope every one of you get into D1. Not gonna lie. All right, D1, D2, D1, D2. All right, so let's cross. So 1, 1, 1, 2, 1, 2, 2, 2. So the chance of getting a palmetto is 50-50. But they asked for the ratios. What was the ratio? That was 2 to 2. That's the ratio. You have 50% of getting a palomino, and obviously 50% of getting a non-palomino. So let's do multiple alleles, and then I'll end it here because it's a pretty long class. Yeah. So all right, we'll do a blood type because covers two topics. I like it. So I will try to fit in the lethal genes in the next class at the start, but yeah, whatever. I will try to fit it in. So let's read. In a human blood group, what will be the blood type of a children when the parents are homozygous type A? Okay, so remember what I said about type A? You write it like this. I know it's a lot of work, and I hate it too. And then here it's just I. Yeah, it's a lot simpler. So now let's cross it. This is A, by the way. So the left side can only pass an IA because these two are the same thing, and the right side can only pass an A. So I. That's the only possibility because this is not even a cross. This is not I can't even make a table out of this. So let's write A. The blood type of the child will 100% be heterozygote type A. So people, if you have some, I know blood types are really confusing because I hated it when I did it. So if you have some question about this, please ask. It's really confusing, I know. All right, type B. Homozygous. Oh, okay, it's just the same thing, guys. So B times I. So what does this mean? Well, you can only pass the I, B, and G. Okay, I'll try writing with the mouse. Yeah, mouse doesn't work. Okay, so what's the chance of the children's gonna be? I, B, I. That's the only possibility. No more. So the blood type of the child will 100% be heterozygote. No, I wrote it wrong. Heterozygote type B. So yeah, these types of questions are usually simple when I do it. So when you guys actually do it yourself, it gets a lot complicated from there. I want you all to understand that, hey, it's not usually this easy. So blood types, they are pretty hard. I know, yeah, they are pretty hard. So always pay attention to this and write as much as you can, because I know it's a lot of work, but when you write it out, it's going to be a lot clearer. And, do I want to do another class? Do I want to do, okay, I will fit lethogene and polygenic in the next class.